Hello everyone, my name is Sneha. I am a student of BSc Chemistry Honours 6th semester. Today I will make a video for visually impaired students on the topic drugs related to chemistry in everyday life. By now you have learned the basic principles of chemistry and also realized that it influences every sphere of human life. The principles of chemistry have been used for the benefit of mankind. Think of cleanliness, the materials like soap, detergents, household bleaches, toothpaste, etc. will come to your mind. Look towards the beautiful clothes. Immediately, chemicals of the synthetic fibers used for making clothes and chemicals giving colors to them will come to your mind. Food materials, again a number of chemicals about which you have learned in the previous unit will appear in your mind. Of course, sickness and diseases remind us of medicines, again chemicals, explosives, fuels, rocket, propellants, building and electronic materials, etc. are all chemicals. In this unit, we shall learn the application of chemistry in three important and interesting areas, namely medicines, food materials, and cleansing agents. Drugs and their classification. Drugs are chemicals of low molecular masses 100 to 500 U. These interact with macromolecular targets and produce a biological response. When the biological response is therapeutic and useful, these chemicals are called medicines and are used in diagnosis, prevention, and treatment of diseases. If taken in doses higher than recommended, most of the drugs used as medicines are potential poisons. Use of chemicals for therapeutic effect is called chemotherapy. Classification of drugs. Drugs are classified mainly on criteria outlined as follows. On the basis of pharmacological effect. This classification is based on pharmacological effect of the drugs. It is useful for doctors because it provides them the whole range of drugs available for the treatment of a particular type of problem. For example, analgesics have pain-killing effect. Antiseptics kill or arrest the growth of microorganisms. On the basis of drug action, it is based on the action of a drug on a particular biochemical process. For example, all antihistamines inhibit the action of the compound histamine, which causes inflammation in the body. There are various ways in which action of histamines can be blocked. You will learn about this in section 16.3.2. On the basis of chemical structure, it is based on the chemical structure of the drug. Drugs classified in this way share common structural features and often have similar pharmacological activity. For example, sulfonamides have common structural feature given below. On the basis of molecular targets, drugs usually interact with biomolecules such as carbohydrates, lipids, proteins, and nucleic acids. These are called target molecules or drug targets. Drugs possessing some common structural features may have the same mechanism of action on targets. The classification based on molecular targets is the most useful classification for medicinal chemists. Drug target interaction. Macromolecules of biological origin perform various functions in the body. For example, proteins, which perform the role of the biological catalysts in the body, are called enzymes. Those which are crucial to communication system in the body are called receptors. Carrier proteins carry polar molecules across the cell membrane. Nucleic acids have coded genetic information for the cell. Lipids and carbohydrates are structural parts of the cell membrane. We shall explain the drug-target interaction with the examples of enzymes and receptors. Enzymes as drug targets. Catalytic action of enzymes. For understanding the interaction between a drug and an enzyme, it is important to know how enzymes catalyze the reaction. In these catalytic activity, enzymes perform two major functions. The first function of an enzyme to hold the substrate. Substrate binds to the active site of the enzyme through a variety of interactions such as ionic bonding, hydrogen bonding, van der Waals interaction, etc. The second function of an enzyme is to provide functional groups that will attack the substrate and carry out chemical reaction. Drug enzyme interaction. Drugs inhibit any of the above mentioned activities of enzymes. These can block the binding sites of the enzyme and prevent the binding of substrate, or can inhibit the catalytic activity of the enzyme. Such drugs are called enzyme inhibitors. Drugs inhibit the attachment of substrate on active site of enzymes in two different ways. Drugs compete with the natural substrate for their attachment on the active sites of the enzymes. Some drugs do not bind to the enzyme's active site. These bind to a different site of enzyme, which is called allosteric site. Thus, binding of inhibitor at allosteric site changes the shape of the active site in such a way that substrate cannot recognize it. If the bond formed between any enzyme and an inhibitor is a strong covalent bond and cannot be broken easily, then the enzyme is blocked permanently. The body then degrades the enzyme inhibitor complex and synthesizes the new enzyme. Receptors as drug targets. Receptors are proteins that are crucial to body's communication process. Majority of these are embedded in cell membranes. Receptor proteins are embedded in the cell membrane in such a way that their small part possessing active site projects out of surface of the membrane and opens on the outside region of the cell membrane. In the body, message between two neurons and between neurons to muscles is communicated through certain chemicals. These chemicals, known as chemical messengers, are received at the binding sites of receptor proteins. To accommodate a messenger, a shape of the receptor site changes. There are large number of different receptors in the body that interact with different chemical messengers. These receptors show selectivity for one chemical messenger over the other because their binding sites have different shape, structure, and amino acid composition. Drugs that bind to the receptor site and inhibits its natural function are called antagonists. These are useful when blocking of message is required. There are other types of drugs that mimic the natural messenger by switching on the receptor. These are called agonists. These are useful when there is lack of natural chemical messenger. In this section, we shall discuss the therapeutic action of a few important classes of drugs. Overproduction of acid in the stomach causes irritation and pain. In severe cases, ulcers are developed in the stomach. Until 1970, only treatment for acidity was administration of antacids such as sodium hydrogen carbonate or a mixture of aluminum and magnesium hydroxide. However, excessive hydrogen carbonate can make the stomach alkaline and trigger the production of even more acid. Metal hydroxides are better alternatives because of being insoluble. These do not increase the pH above neutrality. These treatments control only symptoms and not the cause. Therefore, with these metal salts, the patients cannot be treated easily. In advanced stages, ulcers become life-threatening and its only treatment is removal of the affected part of the stomach. Antihistamines. A major breakthrough in the treatment of hyperacidity came through the discovery according to which a chemical, histamine, stimulates the secretion of pepsin and hydrochloric acid in the stomach. The drug cimetidine tegamet was designed to prevent the interaction of histamine with the receptors present in the stomach wall. These resulted in release of lesser amount of acid. The importance of the drug histamine is a potent vasodilator. It has various functions. It contracts the smooth muscles in the bronchi and gut and relaxes other muscles such as those in the walls of the fine blood vessels. Histamine is also responsible for the nasal congestion associated with common cold and allergic response to pollen. Synthetic drugs, bromophenramine of histamine by competing with histamine for binding sites of receptor where histamine exerts its effect. Now the question arises, why do above mentioned antihistamines not affect the secretion of acid in stomach? The reason is that anti-allergic and antacids drug work on different receptors. Tranquilizers and analgesics are neurologically active drugs. These are a class of chemical compounds used for treatment of stress and mild or even severe mental diseases. These relieve anxiety, stress, irritability, or excitement by inducing a sense of well-being. They form an essential component of sleeping pills. There are various types of tranquilizers. They function by different mechanisms. For example, noradrenaline is one of the neurotransmitters that play a role in mood changes. If the level of noradrenaline is low for some reason, then the signal sending activity becomes low and the person suffers from depression. In such situations, antidepressant drugs are required. These drugs inhibit the enzymes which catalyze the degradation of noradrenaline. If the enzyme is inhibited, this important neurotransmitter is slowly metabolized and can activate its receptor for longer periods of time, thus counteracting the effect of depression. Iprionazide and phenylazine are two such drugs. Some tranquilizers are relatively mild tranquilizers suitable for relieving tension. Equanil is used in controlling depression and hypertension. Derivatives of barbituric acid like veronal, amytal, nembutal, luminal and seconal constitute an important class of tranquilizers. These derivatives are called barbiturate. These are hypnotic, that is sleep producing agents. Some other substances are tranquilizers like serotonin. Analgesics reduce or abolish pain without causing impairment of consciousness, mental confusion, incoordination or paralysis or some other disturbances of nervous system. These are classified as follows: non-narcotic, non-addictive analgesics. Aspirin and paracetamol belong to the class of non-narcotic analgesics. Aspirin is the most familiar example. It inhibits the synthesis of chemicals known as prostaglandins, which stimulate inflammation in the tissue and cause pain. These drugs are effective in relieving skeletal pain such as that due to arthritis. These drugs have many other effects such as reducing fever, antipyretic and preventing platelet coagulation. Because of its anti-blood clotting action, aspirin finds use in prevention of heart attacks. Narcotic analgesics, morphine and many of its homologs, when administered in medicinal doses, relieve pain and produce sleep. In poisonous doses, these produce stupor, coma, convulsions and ultimately death. Morphine narcotics are sometimes referred to as opiates since they are obtained from opium poppy. These analgesics are chiefly used for antimicrobials. Diseases in human beings and animals may be caused by a variety of microorganisms such as bacteria, virus, fungi and other pathogens. An antimicrobial tends to destroy, prevent development or inhibit the pathogenic action of microbes such as bacteria, antibacterial drugs, antifungal agents, antiviral agents or other parasites, antiparasitic drugs selectivity. Antibiotics, antiseptics and disinfectants are antimicrobial drugs. Antibiotics, they are used as drugs to treat infections because of their low toxicity for humans and animals. Initially, antibiotics were classified as chemical substances produced by microorganisms, bacteria, fungi and molds that inhibit the growth or even destroy microorganisms. The development of synthetic methods has helped in the synthesizing some of the compounds that were originally discovered as products of the microorganisms. Also, some purely synthetic compounds have antibacterial activity and therefore the definition of antibiotic has been modified. An antibiotic now refers to a substance produced wholly or partially by chemical synthesis which in low concentrations inhibits the growth or destroys the microorganisms by intervening in their metabolic processes. The search for chemicals that would adversely affect invading bacteria but not the host began in 19th century. Paul Ehrlich, a German bacteriologist, conceived this idea. He investigated arsenic-based structures in order to produce less toxic substances for the treatment of syphilis. He developed the medicine artemisinin known as salvarsan. Paul Ehrlich got Nobel Prize for Medicine in 1908 for this discovery. It was the first effective treatment discovered for syphilis. Although Varicin is toxic to human beings, its effects are on the bacteria which causes syphilis is much greater than on human beings. The arsenic double bond arsenic linkage present in arsenophenamine resembles the N double bond N linkage present in azodides in the sense that arsenic atom is present in place of nitrogen. He also noted tissues getting colored by dye selectivity. Therefore, Ehrlich began to search for compounds which resemble in structures to azodides and selectivity bind to bacteria. In 1932, he succeeded in preparing the first effective antibacterial agent, Prontosil, which resembles in structure to the compound salvaricin. Soon it was discovered that the body Prontosil is converted to a compound called sulfa-anilamide, which is a real active compound. Thus, the sulfa drugs were discovered. A large range of sulfonamide analogues were synthesized. One of the most effective is sulfapyridine. Despite the success of sulfonamides, the real revolution is antibacterial therapy began with the discovery of Alexander Fleming in 1929 of the antibacterial properties of the penicillium fungus. Isolation and purification of active compound to accumulate sufficient material for clinical trials took 13 years. Antibiotics have either cidal killing effect or static inhibitory effect on microbes. A few examples of two types of antibiotics are bactericidal and bacteriostatic. The range of bacteria or other microorganisms that are affected by certain antibiotic is expressed as a spectrum of action. Antibiotics which kill or inhibit a wide range of gram-positive and gram-negative bacteria are said to be broad-spectrum antibiotics. Those effective mainly against gram-positive and gram-negative bacteria are narrow-spectrum antibiotics. If effective against a single organism or disease, they are referred to as limited spectrum antibiotics. Penicillin G has a narrow spectrum. Ampicillin and amoxicillin are synthetic modifications of penicillins. These have broad spectrum. It is absolutely essential to test the patients for sensitivity allergy to penicillin before it is administered. In India, penicillin is manufactured at the Hindustan Antibiotics in Pimpri and in private sector industry. Chloramphenicol, isolated in 1947, is a broad spectrum antibiotic. It is rapidly absorbed by the gastrointestinal tract and hence can be orally given in case of typhoid, dysentery, acute fever, certain form of urinary infections, meningitis and pneumonia. Then antiseptics and disinfectants. Antiseptics and disinfectants are also the chemicals which either kill or prevent the growth of microorganisms. Antiseptics are applied to the living tissues such as wounds, cuts, ulcers and diseased skin surfaces. Examples are furacin, soframycin, etc. These are not ingested like antibiotics. Commonly used antiseptic, Dettol, is a mixture of chloroxylenol and terpineol. Bethionol, the compound is also called bethionol, is added to soaps to impart antiseptic properties. Iodine is a powerful antiseptic. Its 2 to 3% solution in alcohol water mixture is known as tincture of iodine. It is applied on wounds. Iodoform is also used as an antiseptic for wounds. Boric acid in dilute aqueous solution is weak antiseptic for eyes. Disinfectants are applied to inanimate objects such as floors, drainage system, instruments, etc. Same substances can act as an antiseptic as well as disinfectant by varying the concentration. For example, 0.2% solution and sulfur dioxide in very low concentrations are disinfectants. Anti-fertility drugs. Antibiotic revolution has provided long and healthy life to people. The life expectancy has almost doubled. The increased population has caused many social problems in terms of food, resources, environmental issues and employment, etc. To control these problems, population is required to be controlled. This has led to the concept of family planning. Anti-fertility drugs are of use in this direction. Birth control pills essentially contain a mixture of synthetic estrogen and progesterone derivatives. Both of these compounds are hormones. It is known for progesterone suppresses ovulation. Synthetic progesterone derivatives are more potent than progesterone. The estrogen derivative, which is used in combination with progesterone. Chemicals in food. Chemicals are added to food for preservation, enhancing their appeal and adding nutritive value in them. Main categories of food additives are food colors, flavors, sweeteners, fat emulsifiers and stabilizing agent, flour improvers, antioxidants, preservatives, nutritional supplements such as minerals, vitamins and amino acids. Except for chemicals of category nutritional supplements, none of the above additives have nutritive value. These are added either to increase the shelf life of stored food or for cosmetic purposes. In this section, we will discuss only sweeteners and food preservatives. Natural sweeteners such as sucrose add to calorie intake and therefore many people prefer to use artificial sweeteners. Orthosulfobenzamide, also called saccharin, is the first popular artificial sweetening agent. It has been used as a sweetening agent ever since it was discovered in 1879. It is about 550 times as sweet as cane sugar. It is secreted from the body in urine unchanged. It appears to be entirely inert and harmless when taken. Its use is of great value to diabetic persons or people who need to control intake of calories. Some other commonly marketed artificial sweeteners are given in this table. Aspartame is the most successful and widely used artificial sweetener. It is roughly 100 times as sweet as cane sugar. It is methyl ester of dipeptide formed from aspartic acid and phenylalanine. Use of aspartame is limited to cold foods and soft drinks because it is unstable at cooking temperature. Allitame is high potency sweetener, although it is more stable than aspartame. The control of sweetness of food is difficult while using it. Sucralose is trichloro-derivative of sucrose. Its appearance and taste are like sugar. It is stable at cooking temperature. It does not provide calories. Food preservatives. They prevent spoilage of food due to microbial growth. The most commonly used preservatives include table salt, sugar, vegetable oils, and sodium benzoate. Sodium benzoate is used in limited quantities and is metabolized in the body. Salts of sorbic acid and propanoic acid are also used as preservatives. Cleansing agents. Two types of detergents are used as cleansing agents. These are soaps and synthetic detergents. These improve cleansing properties of water. These help in removal of fats which bind other materials to the fabric or skin. Soaps are the detergents used since long. Soaps used for cleaning purpose are sodium and potassium salts of long fatty acids. In this reaction, esters of fatty acids are hydrolyzed and the soap obtained remain in colloidal form. It is precipitated from the solution by adding sodium chloride. The solution left after removing the soap contains glycerol, which can be recovered from by fractional distillation. Only sodium and potassium soaps are soluble in water and are used for cleaning purposes. Types of soaps. Basically, all soaps are made by boiling fats or oils with suitable soluble hydroxide. Variations are made by using different raw materials. Toilet soaps are preferred by using better grades of fats and oils, and care is taken to remove excess alkali. Color and perfumes are added to make these more attractive. Soaps that float in water are made by beating tiny air bubbles before their hardening. Transparent soaps are made by dissolving the soap in ethanol and then evaporating the excess solvent. In medicated soaps, substances of medicinal value are added. In some soaps, Deodorants are added. Shaving soaps contain glycerol to prevent rapid drying. A gum called rosin is added while making them. It forms sodium rosinate which leathers well. Laundry soaps contain fillers like sodium rosinate, sodium silicate, borax, and sodium carbonate. Soap chips are made by running a thin sheet of melted soap onto a cool cylinder and scraping off the soaps in small broken pieces. Soap granules are dried miniature soap bubbles. Soap powders and scouring soaps contain some soap. Why do soaps not work in hard water? Hard water contains calcium and magnesium ions. These ions form insoluble calcium and magnesium soaps respectively when sodium or potassium soaps are dissolved in hard water. These insoluble soaps separate as scum in water and are useless as cleansing agent. In fact, these are hindrance to good washing because the precipitate adheres onto the fiber of the cloth as gummy mass. Hair washed with hard water looks dull because of their sticky precipitate. Dye does not absorb evenly on cloth washed with soap using hard water because of this gummy mass. Synthetic detergents, they are cleansing agents which have all the properties of soaps but these are mainly classified into three categories: anionic detergent, cationic detergent, non-anionic detergent. Anionic detergent, they are sodium salts or sulfonated long chain alcohols or hydrocarbons. Alkyl hydrogen sulfates formed by treating long chain alcohols with concentrated sulfuric acid are neutralized with alkali to form anionic detergent. Similarly, alkyl benzene sulfonates are obtained. In anionic detergents, the anionic part of the molecule is involved in the cleansing action. Sodium salts of alkyl benzene sulfonates are an important class of anionic detergents. They are mostly used for household work. Anionic detergents are also used in toothpaste. Cationic detergents, they are quaternary ammonium salts of amines with acetates, chlorides or bromides as anions. Cationic part possess a long hydrocarbon chain and a positive charge on nitrogen atom. Hence, these are called cationic detergents. Cationic detergents have germicidal properties and are expensive. Therefore, these are of limited use. Non-ionic detergents. Non-ionic detergents do not contain any ion in their constitution. One such detergent is formed when stearic acid reacts with polyethylene glycol. Liquid dishwashing detergents are non-ionic type. Mechanism of cleansing action of this type of detergents is the same as that of soaps. These also remove grease and oil by micelle formation. Main problem that appears in the use of detergents is that slow degradation of detergents leads to their accumulation. Effluents containing such detergents reach the river, ponds, etc. These persist in water even after sewage treatment and cause foaming in rivers, ponds, and streams, and their water gets polluted. These days, the branching of hydrocarbon chain is controlled and kept to the minimum. Unbranched chains can be biodegraded easily. Thank you.